All right, so Biology 1440, lab number 1. The goal of this lab is going to be to show you how to make a figure, which is a graph and caption. The figure shows what your data did in the experiment, and the caption is where you write about what your data did in the experiment. Just a brief description. It's imperative in biology that you think clearly, and you show that you're thinking clearly by writing clearly. Writing is a window into how clearly you can think, and you want to work on expressing yourself as clearly as possible. Now, in biology, when we collect qualitative data, we might write about color change or behavior, things like that. That's one thing. But when we were graphing, we tend to use quantitative data, which are going to be numerical data. And when we use numerical data, we use the metric system. The metric system is based on units of 10, the moving of the decimal place to convert from one unit to another. So, for example, if you're measuring length, you might use meters. Maybe you'll measure in millimeters, which is a thousandth of a meter, or a centimeter, which is 100th of a meter. You might measure in kilometers, which is 1,000 meters. So this shows you how to convert from one form to the other. It's simply a movement of the decimal place, and I assume most of you are familiar with that. I'm not going to do a big lesson on it because there are thousands of lessons online about it, but I will give you a couple of metric conversion questions in an upcoming assignment. So make sure you are familiar with the metric system. Units that we often use in biology are going to include nano, which is a billionth. So for example, a nanometer is a billionth of a meter. I don't use them that much, but that's how you would measure like protein sizes. Micrometers, which has this shape that looks kind of like a U almost, it's called a mu. That's 1/1,000,000th of a meter, and that's how we measure cells, like 30 microns in diameter would be the size of a lot of eukaryotic cells. Millimeters, of course, I'm sure you're familiar with. Meters, I'm sure you're familiar with. Kilometers, so on and so forth. But in biology, we often talk about micro and milli, and the base units like meter. So familiarize yourself with a few of those symbols because you'll see them used in your book quite a bit. Your book also has some information on the metric system in the appendices in the back that you may find useful, as well as information on graphs. Now, when we make graphs in biology, we're going to do all of our graphs this semester will be done on Excel. If you don't have Excel, please download it from the University IT website. It's free for students. University IT has it. Information Technologies is what IT stands for. And when you make a graph, it's going to have an X and Y axis, as we see labeled here. The X axis is where your independent variable goes every time. Independent variable goes there. That is the thing that you have control on changing. For example, how many minutes you exercised, you have control on. The dependent variable goes on the Y axis, and it depends on the independent variable. So heart rate would depend on minutes of exercise. So heart rate would go on the y-axis. And that's how you're going to set up every graph, probably in your entire biology career. Now, let me skip this slide here. So what we're going to do in biology is we're going to collect a lot of data, or you'll be given a lot of data. But you don't necessarily graph every data point you're given. You summarize data. You average data together. And so when you're given a lot of data, you're going to often average it together. And pretty much every graph you'll make in Bio 1 is going to involve averages unless you're told otherwise. And so you may have heard of mean, median, and mode before. We're going to use mean a lot. Mean is the statistical way to talk of an average. So the mean here is what we'll use quite a bit. I don't make much use of mean, median, or of medians and modes, I should say, in Bio 1, but they are useful statistics to be familiar with. But mean is what we will use. So never turn in a graph where you haven't calculated averages, because you've probably done it incorrectly. All right, and so if you forget, mean is the average of the data points. Now, when we make graphs, a lot of times in Bio 1, we make scatter plots, okay? And this one is showing you one drawn on graph paper. I should say line graphs, but we use the scatter plot feature in Excel to make them. Now, this is a very important image that you should come back to over and over again. I will paste this on Blackboard as a file as well. This shows you all the parts of a completed figure. This shows you the X and Y axis that are both labeled, and they both have units on those labels. We can see that there are data points graphed here with error bars. You're going to do that as well. We have a caption beneath. That caption always starts with a figure number. It has a title sentence. We never put titles above graphs in biology. The title goes into the caption beneath the graph. And the second sentence here is a trend sentence, or a sentence that describes the major changes that occurred on the graph. So in this case, we talk about the trends down here and the trends up here. By trend, I don't mean the changes between every data point. I mean, if you were going to describe the major things that happened on the graph, we write about that. And we put a number value of how much change there was on that graph. And that's something that you can do, and you'll see in videos in this lab this week. Another thing that goes on the end of the caption is a statement here that tells the reader what these dots represent. In this case, their mean or averages. These error bars are standard error bars, so it says that right there, and it tells them the sample size. All of this will be explained in upcoming videos, but this is a good figure to come back to look at when you're trying to figure out if you've done a figure correctly. So that is what we're going to do today. We're going to work on graphing. We're going to make column graphs, we're going to make scatter plots, and then you'll have an assignment that goes along with that.